During prophase 1 of meiosis, homologous chromosomes pair up very closely, and corresponding parts of two non-sister chromatids may trade places. This process of crossing over creates variation by producing chromosomes that combine the genes inherited from two parents. Here the process produced a total of 4 genetically different gametes. There are many ways crossing over can occur. In humans, crossover events happen an average of 2 or 3 times per chromosome pair, greatly increasing the variation among eggs and sperm. Note that crossing over produces some parental gametes with chromosomes like those of the parents, and some recombinant gametes with a mixture of genes from both sets of chromosomes.
Alright, so now we're going to start talking about how this process of recombination ties in with genetic variation and genetics. Okay, so far, in terms of our discussions about monohybrid, dihybrid crosses, we have kind of waved our hands at crossing over. Alright, but the reality is that it happens. And so you are going to end up with offspring that are not going to look identical to the parent. Right? You're not going to have the expected phenotypic results from a cross always. Right? You may have some differences in phenotype, where one of the phenotypes of one of the parents found on the other offspring exhibiting the other traits. Right? So you'll see the exchanging of genes or traits between individuals, where you get completely new recombinant phenotypes. Alright, so here, crossing over is interesting because it involves determining how close genes are on a chromosome, or if they are even found on the same chromosome. Alright, so linked genes. Linked genes are inherited together. Alright, so that means that genes that are closer together on a single chromosome tend to be inherited together. The further apart genes are from one another, you may see recombination take place where those genes may not be inherited together, or they at least exhibit a higher frequency of recombination, or two genes can be on two separate chromosomes. Alright, so here, let's look at gametes that are produced from two individuals. Alright, so we have our paternal chromosome and we have our maternal chromosome. Now let's assume there's no recombination. Well, that means that our maternal chromosome is going to give us gametes that look like this. Our paternal chromosome is going to give us gametes that look like this. Alright, now, if crossing over takes place, we'll end up with gametes that will look like this and this. Alright, because we have an exchange of genes between the maternal and paternal chromosome. So recall that crossing over occurs during prophase 1 of meiosis. Alright, you need to remember that. Also, do not confuse linked genes with sex-linked genes. Completely different terminology. Linked genes can either be on autosomes or sex chromosomes. Okay? Sex-linked genes are found only on sex chromosomes. Alright, so how exactly does this idea of crossing over tie into linkage, gene linkage? Well, here's the idea. All right, the further apart two genes are from one another. All right, for example, genes A and B, the greater likelihood or chance that recombination will take place. Okay, the closer two genes are to one another, like B and C, the greater likelihood that they will be inherited together, and there's going to be a decreased frequency of recombination. In other words, these genes are inherited together. In order for these genes to not be inherited together is going to be a situation where you have crossing over that occurs that separates the inheritance of gene B and C in this example. Alright, so the further apart two genes are, the higher likelihood of recombination. The closer they are, the lower the frequency of recombination. Alright, so here we're looking at fruit flies. Alright, so we have the wild type fruit fly with a gray body and normal wings. Alright, so this is the dominant phenotype. In this case, we have a wild type true breeding fly. The other parent is a mutant. It's a double mutant. Alright, so it has a black body and it has vestigial wings. Alright, so vestigial wings are non-functional wings in this case. Alright, so this individual is also true breeding. Right, it's homozygous recessive for both traits. Okay, now the resulting offspring of this cross is that you see all wild type flies. Alright, so all the offspring are wild type. Alright, so in this case, this is the genotype of this fly, the product of this cross. Alright, so it's heterozygous for body type, body color, and wings. Alright, so let's do a test cross. Alright, so if you recall what exactly a test cross is. Well, you're mating your unknown fly, okay, with an unknown genotype. Alright, so let's say we're assuming that we don't know what this is. Okay. With another fly that is homozygous recessive for all traits. Alright, so black body, vestigial wing for this example. Alright, so here we get our offspring. Okay, so keep in mind the potential gametes of this female fly. You can have black body, normal wings. You can have black body, vestigial wings. You can have gray body, vestigial wings, gray body, normal wings, gray body, vestigial wings. You can have black body, normal wings. You can have black body, vestigial wings. Alright, so in this case, this is your parental trait. This is also another parental trait of the first generation. These are recombinants here. Okay. So here, your sperm, the male fly that's got a black body and vestigial wings, can only contribute black body allele and vestigial wing allele. Alright. So the resulting offspring, we get wild type. Alright, so gray body, normal wings. We get black body, vestigial wings. Well, this is parental. This is parental. We would expect that. Okay. Now, we also see gray body, vestigial wings. Well, that's a recombinant. And we see black body, normal wings. That's recombinant. Alright, so if these genes are located on different chromosomes, alright, if these genes segregate independent of one another, okay, then you would expect that you would see a 25% chance, or 25% of your offspring would be of each variant type. You have 20% of your fly population of the offspring that are wild type, 25% that are black vestigial, 25% that are gray vestigial, 25% that are black normal wings. But that is not now if the genes are located on the same chromosome and the parental alleles are always inherited together. Alright, so that means that if the body allele and the body color allele and the wing allele are inherited together, you would see a disproportionality. You would see that you would only see your parental phenotypes. You would only see wild type, gray body, normal wings, or you would see black body, vestigial wings. In other words, these genes are linked. Alright, you would not see any recombinants. Alright, because they're close enough together where recombination is more of a unlikely event. Alright, so the result of this is that out of 2,300 total offspring, you get 965 wild type, 944 black vestigial. You do see some, but not a lot, gray vestigial and black normal winged flies. So about 206 of the gray vestigial, 185 of the black normal winged flies. Alright, so a very small percentage of recombination. Alright, so even though two genes are linked, alright, it does not mean that recombination cannot happen. Okay, it's just that depending upon how close they are, it may be a very small percentage. Alright, so here you're looking at this process of crossing over between homologous chromosomes. So linked alleles, linked genes, can be separated by crossing over, forming recombinant gametes. Now, we can use the frequency of recombination, the percentage of recombinants in a population of offspring, as a way of determining how frequently this occurs and whether or not two genes are in fact linked. Alright, so here we have genes A, B, and C on two homologous chromosomes. Alright, your paternal chromosome in blue has copies of the dominant alleles. In this case, they're all uppercase. And here we have our maternal chromosome. They have copies of the lowercase, they're all recessive alleles. And if you have crossing over that takes place, you end up recombinant with recombinant gametes. Alright, so this is parental. This is parental. Because they look exactly like the parent. These two guys here are recombinant because you've had crossing over take place. Alright, so you have big A, big B, little c on this chromosome. You have little a, little b, big C. Alright, so that tells you you've had crossing over take place. So you get recombinants. Alright, so going back to our little fruit fly dilemma. Alright, so this is showing you the cellular events that took place involving meiosis. And production of gametes. Alright, so here, to kind of explain what you saw before, your black body vestigial winged fruit fly. Alright? It's homozygous recessive for both genes. Alright, so it's only going to contribute a black body allele and a vestigial wing allele. Alright, so that's going to be the gametes at the end of meiosis. Okay. Now looking at our F1 hybrid wild type fruit fly that has a copy of the black body allele, has a copy of the gray body allele, copy of the black body allele, copy of the normal winged allele, and a copy of the vestigial wing. Alright, so here you can get a couple of different combinations. Alright. You can get gray body vestigial. You can get gray body normal. Sorry. You can get gray body vestigial. You can get black body normal. And you can get black body vestigial. Okay. Now, this is parental. This is parental. Alright, because that looks like that, and that looks like that. Alright, those are parental. These are recombinant. Alright, because you have gray body, vestigial, black body, normal wing. Alright, those are recombinant gametes, recombinant chromosomes. So here, at the end of this test cross, with this black-bodied vestigial winged fruit fly, alright, you end up with 965 wild type, 944 black vestigial. Alright, you end up with 206 gray vestigial, 185 black normal. Well, to calculate your recombination frequency, what you do is you add together your recombinants, which is your gray body vestigial, okay, which is 206, and then you have your black-bodied normal fly, normal winged fly, which is 185. So you add 206, 185. 206 plus 185 gives you 391. So 391 recombinants out of a total of 2,300. Alright, so to get the total, you just add 965, 944, 206, and 185, and you get 2,300 total offspring. Alright, so you take the difference, or you divide 391 recombinants by 2,300, multiply by 100, and you get 17%. So the frequency of recombination is 17%, which is pretty low. Alright, so this means that since it's so low, that these genes are linked. They're really close to one another on the same chromosome. Alright, here we're looking at two genes. We're looking at gray body and looking at body color, and we're looking at eye color. Alright, so we have gray body, white eyes. We have yellow body, red eyes. Alright, so here the gray body allele is dominant over the yellow body allele. Here, the red eye allele is dominant over the white eye allele. Alright, so here during meiosis, alright, you end up with your gametes that are produced. Alright, so here we can end up with gray body alleles and white eye alleles inherited together, and get yellow body alleles and red eye allele inherited together. Alright, so the genes for body color and eye color are inherited together. They're linked. Okay. Alright, so will the genes undergo independent assortment if they're on the same chromosome? Alright, so the idea here is that linked genes violate the principle of independent assortment, that two different genes assort independent of one another, which is true. Alright, so you would predict, so here we have a female fruit fly that has red eyes and a gray body. Alright, so here, the fact that we have one copy of the dominant gray body allele and one copy of the dominant red eye allele tells you that we have red eyes, gray body. Here on our male fruit fly, we have white eyes, gray body. Alright, so here we have a copy of the gray eye allele, and we have a copy of the gray body allele. We have a copy of the white eye allele here, the little W. Alright, so this is a sex linked gene. Alright, so males are only going to have one copy of the genes. The mother has two copies of the genes, for which she could be homozygous or heterozygous for either of these. Alright, so looking at the possible offspring. Alright, so looking at the male offspring only that are produced out of this cross, you end up with gray-bodied white eyes, which is parental. Gray-bodied red eyes, which is also going to be having gray eyes, or gray body and red eyes, which is again a parental phenotype. Alright, so it's exactly like the male. Alright. Yellow-bodied red eyes is kind of one of the other possible phenotypes that's you would expect to see. Alright, so here you see very similar to parental phenotypes. Alright, for these traits. Alright, so you see about 4,292 gray-bodied, white-eyed flies, 4,605 yellow-bodied, red-eyed flies. You see 86 yellow-bodied, white-eyed, and 44 gray-bodied, red-eyed flies. Right, and these are only the males. Alright, so there's a disproportionality here. You would expect you would see 1 to 1 to 1 to 1, alright, if the genes were not linked. Alright, but the fact that these genes are linked, you're seeing this disproportionality. Right? So there's a favoritism towards the inheritance of these linked genes, and there's reduced levels of recombination. Right? Alright, so here, linked genes do get inherited together unless crossing over takes place. And you have recombination of new genes. So looking at the recombination frequency, alright, so here you're looking at around 1.5 for your recombinants. Alright, so that's really small. So for our parentals, alright, high level recombination. So these genes tend to be inherited together. Now we can use the recombination frequency to develop a genetic map, a linkage map. Whereby we get an idea of the sequence or order of genes on a chromosome. We're not going to get the exact location, but we're going to figure out the order of the genes. How close they are to one another. It's an approximation. Alright, so here, your recombination frequency can be used to calculate map units. Okay? So, a recombination frequency of 1 is 1 map unit. A recombination frequency of 2 is 2 map units. Alright, so map units are a measure of distance or order. Alright, so genes that are far apart, but if they're on the same chromosome, alright, have a recombination frequency near 50%. Or higher. Alright, so this tells you that the genes are so far apart that they tend to not be inherited together. Or it's possible if they're not. Or they're on 2 separate chromosomes. So that they behave as if they're on different chromosomes. If they're on the same chromosome, but they're really far apart from one another. Alright, so here you're looking at a genetic map of different phenotypes for fruit flies. Alright, so you're looking at genes for body color, genes for eye color, genes for the antennae, genes for other variants of eyes, for wings, for different variants of wing structure, and how close these genes are to one another by using the recombination frequencies. Alright, so we're going to zero in on this here to look at the order of certain genes. So we're going to look at body color, eye color, and wings. Alright, so black body, gray body, red eyes, cinnabar eyes, normal wings, vestigial wings. So you're looking at wild type phenotypes and mutant phenotypes. Mutant phenotypes are on the top, wild type phenotypes are on the bottom. Alright, so looking at some genetic crosses, alright, between these types of individuals, you'll see that you have your body color allele, your wing, your eye color allele, and your wings. Alright, so if you go back to this data here, Right? These are the map units. Alright, the distance between the genes. Alright, so if you subtract 67 from vestigial wings from the cinnabar eyes, alright, you get 9.5. A map unit distance of 9.5. If you subtract 57.5 from 48.5 between cinnabar eyes and black body, or body color, you get 9%. Alright, so 9 map units. 9.5 map units. 9 map units. Alright, so if you add these together, this tells you, if you look at the difference between these two, alright, the difference between the distance between your wings, the genes for the wings, and the genes for body color is 18.5 map units. By looking at the differences in map units between vestigial wing or wing type and eye color, wing type and body color, eye color and body color, you can figure out the orientation of these particular genes along the chromosome. Alright, so they add up to a total of 18.5. So you know the difference between the body color and the wing type is 18.5 map units. So you know that they're the furthest apart. You know that the eye color gene and the vestigial wing is 9.5 map units. Alright, so that's going to be, it's going to come after the body color gene, so it's in between. And the distance between the body color and eye color gene is 9 map units. Alright, so this tells you the orientation of the chromosomes, orientation of the genes on the chromosome. Alright, so here, the frequency of recombination. Alright, so between two genes. Alright, so here we have singed bristles, eye color, and body color. Alright, so the distance between singed bristles and white eyes is 19.6. Alright, so the recombination frequency between these two genes is 19.6. So this is 19.6 map units. Alright, the distance between yellow eyes and body color is 1.4 map units. This adds up to a total distance between singed bristles and yellow body as 21. Alright, so based on the map units, you can determine the arrangement of the genes along the chromosome. Alright, so here, we're going to construct a genetic map. Alright, so we know the distance between singed bristles and white eyes. We know the distance between singed bristles and yellow body. Alright, so we have two other genes we're throwing in there. Alright, so we have miniature wings and we have ruby eyes. Alright, so the distance between the singed bristles and some other gene is 13.5. We know the distance between this gene here, the orange gene, and white eyes is 6.1. Alright, we know the distance between the blue unknown gene and yellow body is 36.1. Well, here you're given a table of recombination frequency. Well, the distance between yellow body and miniature wings. Yellow body and miniature wings is 36.1. Alright, so this tells you here, this is miniature wings. Here. the distance between white eyes and ruby eyes is 6.1. Alright? Well, that would imply that this is ruby eyes. The distance between singed bristles and ruby eyes is 13.5. That matches up. The distance between ruby eyes and miniature wings is 28.6. So here, 15.1 and 13.5 add up to 28.6. Alright, so that's the distance from miniature wings to ruby eyes. Now, you can also have a situation where you have genomic imprinting in offspring. Alright, so here certain genes are going to be silenced based on which parent passes them off to their offspring. Alright, so this involves methylation of cysteine nucleotides in the DNA for a gene. Alright, so you recall that early on we talked about different types of functional groups. We mentioned that methyl groups are involved in regulation. Well, here we're talking about regulation of gene expression. So all of this has to do with embryonic development. Alright, so here, for example, we're looking at mice. Alright, so here we have a normal IGF2 allele that's expressed on the paternal chromosome. And here on the maternal chromosome, we have a normal IGF2 allele that's not expressed. So you end up with a normal-sized mouse. Right, so this is the wild type. Alright, so let's say we have a homozygous mouse. Alright, but you have a mutant IGF2 allele inherited from the mother. Or you have a mutant IGF2 allele inherited from the father. Well, if it's inherited from the mother, alright, you have a normal IGF2 allele that's expressed, normal size, looks exactly like this one here, alright, because the maternal gene is expressed. The mutant IGF2 allele isn't expressed, alright, from the mom. Alright, if we have a mutant IGF2 allele from the dad, and we have the normal IGF2 allele that's not expressed from the mom, we get a dwarfed mouse. Alright, so IGF2 is a growth factor, and it interacts with a IGF2 receptor. Alright, so the genes for IGF2 and the receptor are imprinted. Alright, so here you have the receptor on from the genes from the mom, the IGF2 gene itself, protein is off. Signaling molecule is off. Genes from the dad, IGF2 receptor off, IGF2 protein on. Signaling molecule. Alright, so here, if you delete the mother's IGF2 receptor gene, the receptor gene produces overly large offspring. Deleting the father's IGF2 gene produces dwarf offspring. Deleting the mother's IGF2 receptor gene and the father's IGF2 gene produces normal-sized offspring. Alright, so here the imprints of IGF2 and its receptor normally cancel each other out. Alright, so changing the imprint on one copy of a gene has an effect on the size of the offspring. So you can either end up with large or small offspring, depending on whether one of the genes is mutated or expressed. Alright, so here, the normal IGF2 gene variant is expressed from the parental chromosome, normal size of the mouse. Alright, the mutant IGF2 gene variant is not expressed from the maternal chromosome. Alright, so in this case, it's not expressed because it's been methylated. Okay. Now, if the IGF2 gene variant from the dad is expressed, you get a dwarf mouse. No IGF2 gene variant not expressed from the maternal, get a dwarfed mouse. Alright, so methylation, you can basically turn on or off certain genes. Alright, so we're talking about the gene for the protein signaling molecule IGF2 and the protein receptor, IGF2 receptor. Alright, so lastly, right, looking at inheritance of organelles. So we're going to focus on extrachromosomal or extranuclear DNA. So we're going to talk about mitochondrial DNA and mitochondrial genes. Alright, so you'll recall that the mitochondrial chromosome is really small. It's circular, double-stranded DNA. Alright, it's very similar to terms of similarities in size of bacterial chromosome. Alright, so here the mitochondrial DNA is actually inherited maternally. Alright, so we inherit our nuclear DNA from our mom and our dad. We inherit mitochondrial DNA from just our mom. Alright, so here this is showing you that nuclear DNA is inherited from all ancestors. Alright, so both our grandparents, grandfather, grandmother, alright, on both sides of the family, our parents, our parents' parents. Mitochondrial DNA is matrilineal. So a single lineage from the mom's line. So your mother, your mom's mom, so your grandmother, your grandmother's mother, and so on. It's a matrilineal lineage. Now, here you have your mom, you have your dad. The mom's nuclear DNA in the egg, the father's nuclear DNA with sperm, given their haploid fused together, form your chromosomal DNA, but your mitochondria. Your mitochondria come from your mom. Now, what about individuals that have mitochondrial disorders? Alright. So there's a way around that. Alright, so this particular procedure is being tested in Britain. Alright. United States is looking at possibly approving the procedure. Alright, so here, what you have is an egg with abnormal mitochondria, with normal, healthy nuclear DNA from the mom. So what you do is you remove the mitochondria, and you're left with the maternal nuclear DNA from the mom. You have another healthy female egg donor. Alright, she has healthy mitochondria. You discard her nuclear DNA, but you keep her mitochondria. You fuse together, you insert the nuclear genome into the oocyte with the healthy mitochondria. Alright, and then you use your father's sperm to inseminate the egg through in vitro fertilization, and then you have a developing embryo that has no mitochondrial diseases. Okay, so here you would have an individual that has three parents, all right, mom and dad through nuclear DNA, and a third parent for mitochondrial DNA.